For questions during Q&A, you can either raise your hands through the reactions option or type it in the chat. Without further ado, let me introduce to those who are joining us for the first time today what Plant.ID and this seminar series is about. Plant.ID is a Marie Curie ITN network which kicked off in 2018 with 21 beneficiaries and 23 partner organizations. We are a diverse array of researchers coming together to molecularly identify plants. This seminar series is the collective showcase of the work done by the 15 PhD fellows in here, myself included, where over the course of 5 sessions, we get to hear a little bit more about each of us. In the last 6 months, we have heard from 6 PhD fellows, and today, we are already at the halfway mark of the seminar series. The topic today is on proxy wars, molecular tools for reconstructing diet and environment. So before we start on the talk, I'll give a short background about myself. My PhD research is on the molecular analysis of feces to study the diet of the Western capercaillies. For those of you who are unfamiliar with the bird, you can do a quick YouTube search on David Attenborough and capercaillie attack to get a sense of the gentle nature of these birds. Since today is Earth Day and unfortunately we cannot travel, our talks will be a little special where our speakers will bring you traveling back in time from the Pleistocene through the Holocene period and ending in modern times. Here is today's program where there will be 2 student talks followed by a short break, and we will end the session with 2 keynotes. So now we will move on to our first student speaker, María Ariza. She did her bachelor studies in biology at the University of San Carlos, Guatemala. For her master's, she was awarded the Erasmus Mundus Scholarship in Applied Ecology. There, she studied the interactions between plants and bats in the neotropics. Currently, she is a PhD student based at the University of Oslo, and her research is about ground-truthing soil eDNA from molecular vegetation assessment. Besides her PhD, she is also actively involved in science communication and is currently preparing a Plant.ID outreach campaign that will soon be launched. I'll now hand over the time to Maria.
Thank you, Physilia. Going to share my screen now. Yes. So yeah, let's start. Today, as we mentioned before, is Earth Day. And I would like to start to talk about the Earth's most predominant life form, which are plants. While we only know 8% of its expected diversity, around 400,000 known species, plants are useful for human societies and ecosystems. And derived from this extensive use, as you can see, we have here like the number of species that serve for different purposes. This extensive use has turned out in a threat. Currently, plants are under threat, and the main threats are agriculture, biological resource use, and natural system modifications. Facing these challenges and threats, plant assessments have never been so important, especially to discover, reveal, and protect diversity. But to assess biodiversity, we employed proxies. And these proxies can be morphologic based or molecular. So traditionally, we have been inventorying plants one by one by expecting morphological characters. And nowadays, we can use molecular tools and samples that harbor DNA, for example, an environmental sample that can be soil, water, or air, or analyze completely complete genomes, yes. And, but if we zoom into these 2 types of proxies, morphology and molecular proxies, little we know about how these 2 compare. And in other words, in the proxy war, who will win? And particularly for those proxies, molecular proxies using soil eDNA, little we know about the ground truth of the soil eDNA, meaning that we don't know for sure how the plant DNA signatures in the soil relate to the vegetation. In addition, we know from previous studies that soil harbors spatial and temporal signals. From studies in tundra soils, we know that the soil is signaling mainly those plants that are near to where the soil sample is taken. And also from cultivated soils, we know that soil, it's also signaling past vegetation. So there is a temporality and implied. So to deal with this lack of knowledge, we designed a study in which these two proxies are compared. And for this, we are comparing a vegetation assessment that was carried through morphological inspections and in 100 plots that are 1 meter square. And these are located in across spruce and pine forest in the Solholmfjell forest, which is in the south of Norway. And 7 observations were made across 30 years, and each observation spans 5 years, as you can see. And across the 7 observations, these 30 years of assessment, it was 156 taxa was assessed. Then, to compare these assessments to the one carried by soil eDNA, we took a soil sample in 2018 at the center of each plot, and we took the topsoil, mainly the organic horizon. Then we amplified the TRNL P6 loop, and we carried 3 PCR replicates per plot, and we added 12 negative controls. And also, it must be said that TRNL could only detect 121 taxa from those 156 recording on the morphology base, because we only had, there is only a reference sequence for this amount of taxa. That means that we have around 35 taxa that are undetectable by TRNL. And yeah, so let's see who will win in this proxy war. So when we take a look into how soil eDNA is doing, the performance of this proxy, you can see here the number in blue, you can see the number of taxa that couldn't be detectable by TRNL, as I said before, those 35 species, right? And then in the red or pink, you could see those plants that could have been detected, but they were not detected. And in green, the plants that eDNA find and that were matching to that plot, that were expected into that plot. So, and we divided this analysis between those plots that are in the pine forest in respect to those in the spruce forest, as you can see. And first of all, you can see that we have like more species on the spruce forest, either like expected or detected by DNA. And then here we also see that we did not recover everything that it was expected on the plot. And then when we see the match, the green part, we see that we could think that they are differences between the pine forest and the spruce forest, but when we assess differences regarding the match by the forest type, we didn't find any differences, suggesting that the match is not underlined by the plot type. And now, however, but if we go and see again to the performance of this proxy, as we saw before, we didn't find everything that it was expected. However, we found more than it was expected, because here I have the same information, just in the section B, in the plotted down, you have it in the proportion. And then here again, we can see the number of taxa that match to regional flora. This is in purple, and this means that these species were not expected, but also are expected to be found in the region, in the Solholmfjeld region where these plots are. And also in blue, we have the number of taxa that was not expected within the plot, but it was expected in other plots, right? And again, the same information in green and in red. And then this graph is showing us that soil eDNA, it's providing more information than expected, although that the match is low, but still we are lacking some species that could have been detected, right? And now, when we go and ask what is the temporal resolution of a soil sample, what is the proxy resolution in terms of the time scale? So we calculated the mean match to one of each of the years that the assessment was carried. So that's what you have in the x-axis. And then we compare this match, and then we see that the greatest match is to that vegetation that was assessed in the year that the soil sample was taken, so in the year 0, and a little bit as well of of the flora that existed 5 and 10 years ago. And in the letters here is just plotted what the comparison, the pairwise comparison across each of the assessments. And then if you have a shared letter between two groups, that means that there is no significant differences. And then if you have different letters, that means that you have significant differences. So here, yes, we can see that the temporal resolution of soil eDNA, it's about mainly the current vegetation and a bit of the past. It's giving us a little bit of a blast from the past. And then regarding those species that matched, we asked, okay, what it's underlying this detectability? When we detect something that we expect in the plot with soil DNA, what it can be, it's because those plants that were detected, in this case the match, on the x-axis is because they are highly abundant. So we retrieved the abundance from those plants that match and didn't match. And here in this violin plot, you can see that here is the distribution of the data that is plotted. So there is in the no match in the left, you have a lot of species with low abundance, whether in that wide of the match on the right, you have a lot of species that have higher abundance. And when we compare this, we find that there were significant differences in abundance between those plants that match and didn't match, suggesting that abundance is underlying detectability. And with that, I would like just to tell you a few take-home messages. That first, soil eDNA recovers higher plant diversity than expected by morphology-based proxies. That soil eDNA signals present plant biodiversity and a bit of a blast from the past. And that soil eDNA signals mainly abundant plant biodiversity. Now I would like just to finish here with a little bit of self-promotion. We are also comparing soil DNA assessment to typification of nature types. Yes, stay tuned to see how this proxy war will turn out. And if you want to know a little bit more about soil and assessments, we stay tuned for these 2 chapters that I am leading with Sandra Garcés Pastor, one of my co-authors and the speakers for today. And the book is coming out soon this year. So yeah, stay tuned, and thank you very much.
Okay, thank you, Maria, for the very interesting talk. Can I see if anyone has some questions? So maybe Maria, you have to stop sharing your screen for a while. So if you want to ask some questions, you can ask through the chat option or just raise your hand using the reaction button. Okay, so I have one question. Because you detected like DNA from the past, I was just wondering how can DNA persist in soil for such a long time, given that they degrade immediately when it's shed from the plants?
Yeah, and it can also degrade a lot because there's a lot of decomposition going on in the soil, and that's a good question. Yes, so soil eDNA persists in the soil because it can the DNA can attach to clay particles. So when DNA attaches, then it can it's more robust and it can stay for longer. And also DNA can be ingested by some bacteria and in something that is called horizontal gene transfer, and then in this way it can be integrated and remain more. And also, there is a lot of process in the soil that are turning the DNA, right? So all of that, the biota that is moving underground, it's also like bringing up DNA that is and mixing it, right? So yeah, it can persist in different ways.
Okay, thank you for your answer. Is there anybody with another question? Okay, if not, then we will now move to our next student speaker. So our next student speaker is Marcel Polling, who is a PhD student at the Naturalis Biodiversity Center in Leiden. He studied geology in the Netherlands before working for 5 years as a professional palynologist in North Wales, United Kingdom. For his PhD, he is using the fossilized remains of pollen and algae to determine the age and paleo environment of million-year-old rock samples. So Marcel, the floor is now yours.
So very sorry, I just got someone at the door with a package for me. Very good timing. Okay, right, here we go. So yeah, thank you for introducing me, Physilia. My name is Marcel Polling, and today I'll be talking about the hidden biodiversity of pollen. And I presume we are all biologists here, so a lot of you will be thinking, well, pollen, they're not really hidden. I can just put them under the microscope. And I can see them, and I can see there's different types, so I can just color them and see that the large green balls here, they must be from the same species or the same family, and in that way I can distinguish them. But actually, there's a lot more going on that you can't see. So here's an example from the nettle family under a light microscope. I've put some pollen grains here. And image them, and as you can see, they look very similar, almost alike, but actually they're from many different species from this nettle family, and even from some different families. So this one is, for example, from Cannabaceae and the Moraceae. So actually pollen from different families even make pollen that look very similar and that you cannot distinguish. But actually it is important to distinguish them because, for example, if you look for hay fever forecasts, then there will be pollen grains that are non-allergenic versus pollen that are severely allergenic, but you cannot distinguish them if you look down the microscope. What's more, you also wouldn't be able to tell if the pollen is coming from native species or is it coming from species that have been introduced. So what is the contribution of exotic species, for example, to pollen in the air? But apart from that, you also have no information about the paleo environment or the environment that you could reconstruct using these pollen grains. So actually there's so much information missing if you just look at them down the microscope. Of course, I've only shown you here the one example. This is the nettle family, but it counts for nearly all pollen families. It's very hard to distinguish species. So yeah, that's basically my PhD. That's what I'm working on. I unravel the hidden diversity of pollen grains. And I use two techniques to do this. I use automatic image detection and molecular techniques using DNA metabarcoding. And today I'll be talking about the DNA methods. So yeah, I try to get DNA from pollen grains to distinguish them to the species level. And I look at ways of quantifying pollen using DNA. So first, I want to talk about a little bit of the background of this project. For hay fever sufferers, now it is 1 in 5 people globally that are suffering from hay fever, and this is increasing constantly. And a lot of studies have shown that this is most likely due to increased CO2. But actually what is, so there's also a recent study actually that I want to maybe point your attention to, that there's a higher airborne pollen concentrations are also linked to increased COVID, but I'm not going to go into that, but maybe interesting for you to have a look at. So questions that I want to answer is, what is the exact species composition of pollen in the air? Are the hay fever symptoms mostly caused by native species? And most importantly for me is, can we quantify? Because it's not really important if we just know if a certain species of pollen is in the air, but how many of them are there relatively or for real. So the material I am working with is collected using this pollen air sampler. It's called a Burkhardt air sampler, and it collects pollen from the air, just sucks it in, puts it on sticky tape, and palynologists just count those pollen down a microscope. You just put this on every day. They simply give all the different identifications, and people suffering from hay fever can have a look at this and say, oh, I suffer from grass pollen, for example, and see that there's a lot of grass pollen in the air at a certain point. So I received the samples from 2 different stations in the Netherlands where that collect these pollen from the air. One is here in Leiden and one in Helmond, near Eindhoven in the Netherlands. And I received 2 different types of material. I received the tape directly from this Burkhardt air sampler, but also the pollen slides. So in total, I've got 60 samples to work with. Well, actually, I had nearly 50,000 samples to work with, because people have been collecting these samples since 1970. But we quickly found out that you cannot get DNA from these pollen samples if they're older than 2 or 3 years, then the DNA becomes too degraded. So we have these samples from 2019 and 2020. So just to run you through, how do you get DNA from pollen grains? Because the DNA is actually locked inside this tiny pollen cell, and to get the DNA out, you actually have to lyse the pollen grains. You have to destroy them to free the DNA. So we use a lysis step using these stainless steel balls and pollen lysis machine, which basically just shakes it very violently for 6 minutes. And then you can see, I checked it under the microscope, you can see that the pollen grains have opened and the DNA is free. So actually from the tape, we just put that straightly into a tube, but from the microscopic slides, we had to open them. So we heated up the slide, open it up, and get the tape out. So then we amplify the DNA. We use the DNeasy plant kit, amplify it, and use metabarcoding technique using an Illumina. So then I amplified two different regions from this from pollen. I used the short marker, the same one as Maria was just telling you about, this tRNL, which is between 8 and 152 base pairs, so very, very short. It's ideal for if you want to work on degraded DNA, but it has a relatively low taxonomic resolution, especially for some groups that are very abundant in pollen, like grasses. And it is plant specific. And then I also use another marker. This is ITS2, internal transcribed spacer. This is a nuclear marker, which is a lot longer, between 400, 500 base pairs. So you need relatively good quality DNA. And so the pro of that is that you have a very high taxonomic resolution. But this marker is actually shared with fungi. So to avoid amplifying fungi, we use plant-specific primers to just focus on plants. So some first results. This is, I will just show a couple of different samples here. This one is from 24th of June 2020. So here in blue on the left-hand side, you can see the pollen counts that have been made manually, so down the microscope. And then here on the right-hand side is the square root of the amount of reads obtained with this tRNL, this short DNA marker. You can see that it finds all the taxa that we expected to find, but that actually if we correlate it, so actually here I log transformed the counts and the reads. And if you correlate them, actually it's not very high, and you can already visually see that sometimes it doesn't really match. But then if we go to this long marker, this long ITS2, we can already see that again we find all these pollen taxa that we expected to find, and actually this correlation of the log transfer abundance versus the counts is a lot higher. But now I'm just showing it for one slide, but I want to convince you that this is what I find consistently for all the material. So here, for example, is another microscopic slide from February 2020. Here we find a lot of alder pollen, alnus, and Cupressus pollen. With tRNL, actually, we find a slightly higher correlation here, but with ITS2, it's higher consistently. So what I keep finding is that ITS2 seems to do a lot better in quantifying the pollen grains. So that's actually a really nice result, because there's a lot of controversy about this. Can you count using DNA? And of course, it's very, very tricky. But these preliminary results at least show that you can probably use it. Now, of course, there's more that you get from getting DNA from pollen. You can also find more taxa than you can using just looking down a microscope. Now, I think it's very important to note here that I used a local reference database. So I only included references in my database from species that are either native to the Netherlands or that are known to be in cultivation or are known to be exotic species. And in that way, using the short DNA, the tRNL, we see that we can, for some taxa, we can get an increased taxonomic resolution. For some we don't, but yeah, it does seem to work well. But now if we look at this longer DNA, this ITS2, we actually see that we can get species level resolution for nearly all our taxa, and we find a lot of species that were not recognized using the pollen, using the microscope. Now, of course, what's even nicer about this is if we have a species resolution is that we can start to say, well, actually there's species here, for example, in this nettle family that are severely allergenic, while for example, this Urtica dioica is not allergenic at all. Parentaria judaica is severely allergenic. So now we can really start to find trends in, and we can see that it's not just this species of Urtica, but also this one. And we see that there's pollen coming from exotic species that are not native to the Netherlands that couldn't be recognized before. So yeah, this is giving us a lot of new information. So to conclude for this section is that, yeah, we can probably use the DNA reads to quantify pollen grains. TRNL and ITS2 don't always give the same results. They actually give complementary results. And we can use this to find trends. And now, as they say, I want to move on to something completely different, because we were talking about what information we get from pollen and what can we learn from them. So for a little change, I thought it would be nice to talk about this. And maybe a lot of people will think, oh, we're going to talk about mammoth having hay fever. But no, I'm sorry, I won't. No, I actually will talk about this landscape that you see over here, because this landscape is reconstructed traditionally using pollen grains. So the way this drawing is made, the artist knows most of it, what he knows, because of people studying pollen. And what they found consistently was lots of grass pollen, lots of types of pollen that were super abundant, and that sort of created this image of this vast grassy plains. But recent studies have already shown that that may be incorrect, actually. So because we're talking about proxy wars today, I also was involved in this study where we used pollen analysis, but also macrofossils and DNA and compared them. So I actually have 11 samples from permafrost. And they are up to 30,000 years old, and we look at the pollen, we look at the macrofossils and the DNA, and we see what information can we get from the three, and how similar is it, or how dissimilar is it maybe. Again, I use this short DNA marker, this TRNL, and I use ITS2, so very long DNA, and I see if we can amplify this. And I found that this long DNA, this ITS, we can actually amplify it from nearly all samples, apart from 3 of them. Then if we look at the comparison, actually what we found is that at the genus level, looking at the plants that we find mostly, most of the unique taxa we find using DNA, and that the overlap is highest between DNA and the macrofossils, which is sort of what you expect, because the macrofossils are in the dung of these samples. So you expect the highest overlap. But still, we find unique genera using the macrofossils and the pollen grains as well. So if you just look at DNA, you don't get a complete picture of everything that's in that sample. So yeah, and finally, of course, I want to integrate all the information that we get from the different proxies to reconstruct the diet and the habitat of these animals. So that's what you see here. So these are the different proxies, the pollen, the macrofossils, TRNL and ITS markers. And I divided all the plant taxa that I found into these groups. There's forbs, which is flowering plants basically, and graminoids, mosses, shrubs, trees, lichen. What we found is that for some samples, these are, for example, mammoth samples, we find that all proxies basically show the same thing. These animals were just eating grass, according to all the proxies. But that's only for these samples, but there were also some of the samples that showed that the amount of DNA is actually showing that the animals were eating shrubs mostly, while the macrofossils were showing something completely different. And for some, for example, here where in this sample where we found 100% grass as macrofossils in this sample, using DNA, we found many more taxa that are probably from flowering plants. And we think that's probably because it's very hard to recognize these taxa as macrofossils, but using DNA, you can really start to see what they are. So to reconstruct the diets of these animals, I actually took the average value from the macrofossils and the DNA. And why did I exclude pollen? That's because the pollen are most likely for more of a regional signal, and while the macro and the DNA are much more local. And in that way, we could see that that even within the animals, for example, I have 5 mammoth samples here, you can see that they weren't all just eating grass. Here, for example, there's one that's clearly eating grass, and this one here maybe as well. But there's a lot of variation there. They're not just eating grass, but all sorts of different taxa and different groups. And again, we don't, so that's more of the quantifying, but qualitative information is also highly increased if we use DNA, of course. So here are 2 samples that are included in this study using the pollen and the macrofossil results. These are the results. So we just find a lot of grass, some Artemisia, and we have sort of semi-quantitative result. But then using DNA, we find this whole wealth of species, and we can get so much more information using DNA out of this. So I don't want to go too deep into this, but the main message is that we got a lot of species level identifications using these DNA. And then we can use that to reconstruct what the habitats may have looked like of these animals. So here are again the 11 samples that are included in my study, and we divided all these species level or sometimes genus level identification into ranging from steppe, very dry, to sort of very wet, to marsh. And then we can start to have an idea of what the habitat may have looked like, where the animal was living in. So we can see for these mammoths, for example, in the Pleistocene, that there was clearly a steppe component. While for these more recent reindeer samples, there's much more of a tundra and Arctic alpine environment. Yeah, so I won't go much deeper into that because of time, but yeah. So thank you very much. I hope to have convinced you that there's a lot more than meets the eye, more information to get from pollen. And if there is any question, please let me know.
Thank you, Marcel, for bringing us back in time and for the very interesting talk. Can I just see who has some questions by raising your hand or in the chat? Okay, so Thibault has a question.
Yeah, just out of curiosity, do you identify so you have the dung, you made metabarcoding on the dung. Do you identify it by the diet of the animal, or do you find some DNA from the animal, some sequences from the animal in the dung, or is it just pollen? Yeah.
Yeah, so for we use primers that only amplify plants. So in that way, we don't find any of the mammal DNA. But okay, for some of the samples, we had to validate that they were actually made by, made, I'm not sure if that's the right word, but produced by the relevant animal, and then we confirmed them using mammal-specific primers.
Okay, yeah. Thanks.
We have a question from Yannick. Yannick asks if you saw any signs of DNA degradation in your sequencing results from hitting the pollen slides as compared to fresh samples.
No, I'm thinking. I don't think I don't think I saw that. No. There will be something to look at.
Any other questions here?
Is
I have
Inge?
no. Okay, yeah, Inge.
I really enjoyed your talk, and I have many questions to both parts, both the pollen and the ancient DNA.
Yeah.
But basically, you expect to find the same thing, the same species, and I wonder to which degree you did that. Because if I look at your seeing your diet analysis, generally the DNA was sort of more similar to each other than it was for the pollen or macro.
Yeah.
But did they really show the same? And how did you get that much graminoids out of the TRNL, which is generally underrepresenting the graminoids?
Yeah. So yeah, I don't know how I did that, to be honest. That's just the results that I found. But I think if we maybe you're referring to those two samples where all the proxies were showing basically 100% graminoids.
Yes.
Yeah. I think for these samples, I think there just literally wasn't much else in that sample, and the macro fossils were also showing basically 99.9% was grass stems and grass caryopsis and seeds and stuff. So I think if you have a sample like that, then you can just get TRNL, and of course with TRNL from grasses you only find maybe maximum to the subfamily level, and sometimes some species, but not so many. So there the ITS marker really showed its value and could really identify the grasses to the species level.
It's the first study I see with that old samples using the ITS. So I'm very curious about how does the species fit with the other proxies from the ITS? Do you think you get just a subset of the species, or do you actually get a representative presentation?
Well, the problem with the macrofossils is that so we only have one validation every time. So macrofossils, they were so they were really degraded because of the chewing action of the animal, and so they were very hard to identify. So most of them were just identified to family level, basically, so just grass. And then if we compare the TRNL and the macrofossil results, then we see that there is a lot of overlap, but at different taxonomic levels. So you do find some species shared between TRNL and ITS2, but a lot of the time, and most of the time, the ITS is showing species where TRNL is maybe showing subfamily or similar.
Okay, yeah, for the grasses, I can see that's a problem.
Yeah.
Okay, thank you.
Yeah, thanks.
Okay, thank you, Inge and Marcel. So now we'll go for a short break, whereas during the break, you'll feel free to type in the chat to ask the speakers more questions if you want to. And please come back here by 3 p.m. so that we can commence our 2 keynote talks. Thank you.
Oh, fuck. I got 3.
Okay, I hope that everyone have had a nice break and that most people should be back by now. So now we'll start with our first keynote speaker, Dr. Sandra Garcés Pastor. Sandra did a PhD at the University of Barcelona, where she compared between using molecular techniques and microscopic analysis to identify pollen and diatoms. For a postdoc, she's currently based at the Arctic University of Norway in Tromsø, where she's working with sedimentary DNA to disentangle climatic and human pressures on vegetation communities. I'll now hand over the time to Sandra.
Thank you very much, Physilia, for introducing me. So I will talk about longer time scale as the samples that Maria presented, not so long as some of the big mammals that Marcel talked about or he presented. But I just wanted to show this introductory slide that it's about paleoecology. And as you know, and we have said that today it's the Earth Day, you know that all the ecosystems are changing at they are changing very quickly. And one of the problems about the modeling of how this climate change will impact our ecosystems, it's a very limited instrumental data, like more or less 100 years if we go further. So paleoecology allows us to reconstruct the organisms that were in the past and reflect how those organisms responded to the past climate changes and human pressure. And then all this information allows us to make better models to infer the impacts of the current climate changing. For that, paleoecologists usually use the sediments from alpine lakes or high mountain lakes, which are less influenced by humans, and also recover all the sediments and the material that comes from the catchment to the lake. So it is very well sedimentated a long time. For that, we use several indicators like pollen or charcoal to infer fires, algae to infer what happened inside the lake, and tree rings. But we can also use DNA to check how to reconstruct all those organisms and see how they responded to climate. The study that I will present is inside the EcoChem project that wants to see the effect of climate, biota, and humans in the alpine species of two different regions. You can see that we have northern Norway and also the Alps. They share alpine taxa, but they have a very different human impact history. So we have two scenarios of similar taxa responding to the same climatic pressures a long time. So we can see changes in the species richness and species pool. In this project, we also did the two big reference databases for Philo Norway and Philo Alps from herbarium and nowadays material, reaching a high amount of taxa that are locally from the regions that we are going to study. Now, I will focus on the lakes of the region of the Alps. These are some of the lakes that we sampled during fieldwork. We already extracted the DNA and sequenced, and here you can see some of them. And today I will talk to you about Suusuli, the one here in the Central Alps. The reason is that when we check all the taxa of these lakes, Suusuli, it's the most diverse of all the studied lakes that we have been studying so far. So we will center into this one. This lake is located in the subalpine stage, almost 2,000 meters above the sea level. As you know, vegetation can organize according to altitude. So temperature makes change the community, the species communities that we can find. And then you have very characterized taxa in each altitudinal stage, which makes it very easy to see and imagine in what stage you are. And also, if you change your temperature, these communities will expand or shrink. And then we can see how this vegetation change to past climatic changes. So the main question of this study is what are the vegetation responses of Sulsuli to climate and humans along the Holocene? For that, and also what ecological inferences can we get from this data? For that, we used a multi-proxy approach. So we used some proxy from some NPP or coprophilous fungi that you can find in the feces of animals, and they will give us, they are an indicator of cattle or grazing. We will also use the chironomids to reconstruct the past temperature changes. And the same, we will also use charcoal to reconstruct fires that could have occurred in the past and also affected our vegetation. Of course, we will use the DNA to reconstruct the vegetal communities and the richness of those communities a long time. So to talk a bit about material and methods, we extracted the DNA using a DNA Easy Power Soil kit. We used the same marker as Maria and Marcel used the tRNL P6 loop and 8 PCR replicates. And after the sequencing, we compared our sequences with the PhyloAlps database with more than 4,000 species for covering all the alpine flora, all the Alps flora, and also 3 more databases, Philo Norway, that is covering the flora from northern Norway and Norway, Arbor-Brajo, EMBL. We also took some macro remains that allow us to do an 8-step model. So you can see that the time that covers our the record that I'm going to present you, it's 11,000 years old. We get to a deeper, we have like 1 meter of sediment that we also extracted DNA from, but just take in mind that this part is extrapolated, so there might be there might be a bit uncertainty about the proper age of those of this part of the sediment. So far, the 8-step model looks very good and it makes a lot of sense. This is 3 different parallel cores that were extracted from the lake and were correlated between them. Here you can see the vegetation community covering 376 taxa between trees, shrubs, forbs, ferns, and aquatics. From this taxa, 146 get to genus level, 140 to species level, and the others are at a higher taxonomic resolution. Here you can see all those taxa aggruppated according to the functional groups, where you can see the proportion of the relative proportion of the reach of each of these groups. But I won't take much time in this sample. In this slide, I will go for the others. So here you can see the for those who are familiarized in a paleo-ecological diagram, you can see the depth, the age, and the coprophilous fungi, the charcoals, the main functional groups, together with the taxonomic richness the temperature reconstruction, an average to see the quality of those of the DNA of those samples and the figure that we already saw. I will also be fast with that one, but you can see a main transition from the early Holocene to the mid-Holocene, from the herbs to shrubs and trees, and another phase where we come back to meadows and then to trees and back again to meadows. And you can also see at first sight a lot of changes in richness along these 11,000 years. And as I told you before, vegetation organized according to altitudinal beds. So when we take a closer look to all the taxa that we find in this community, in this vegetation, you can find that some of those taxa can be related to different altitudinal stages or zones. Then, if we can manage to reconstruct those groups of taxa that belong to different stages, we might see how they change according to past temperature changes. And we have the temperature from chironomids from the very same lake. So we can see which are the vegetation responses in the past. Then how do we do that? What we did is that from these 376 sequences, we applied like an autoecological filter, which is like we use some guides that they already know the autoecology of those species. And then we compare to them. Of course, there are some species that are very cosmopolite. So they can live in several altitudinal zones, and then they are not useful to discriminate which was the zone that we have in the past. For those sequences that we got species taxonomic resolution, it was very straightforward. You have one species, go to the guide and find it, and then you see what it's telling you. But I will put here two examples that you can also find when you share your sequences with the taxa, with the databases. So for this sequence, you have 4 different species that if you have to end up with one group with what name for that sequence, you will end up with a family level, because as you can see, they belong to different genus. But there is another way to approach to this taxa. So all these species that share the same sequence, we can look at the autoecology of each of those species. And then we can give a value to each of them. For example, in that sequence, the 4 species give 2 different altitudinal information. So that would be a bad indicator. Let's see the other example, where we have 3 different species that share the same sequence. And if we had to get some information about, so if we approach with that species autoecology based thing, for example, in this, we see that all of them give us the same value. 2.5 would be montane subalpine taxa. Then that could be a good indicator for these altitudinal stages. Then we end up with 140 sequences, and we checked if it was coherent to find those sequences in the region of our study site. And we end up with some reliable indicators covering from montane to alpine belt. We did the same for arable indicators and for pastoral indicators, and I will come to them later. So we start with this functional group diagram, and here we have the altitudinal group diagram. Let's have a closer look to this one. So the first stage, what we can see, that this is older than 11,000, so it could be, if we, this is the part of the age-step model that it was a bit extrapolated, but it could be around 12,000 years ago. You can see a predominance of alpine type taxa. Here you also have a small summary of all the vegetation that we find, and some vegetation that can live in two altitudinal zones. Okay, so they are according to this. You also have the temperature, richness, functional groups, and some erosion indicators. And LOI, it's a organic matter indicator. So we can see that we have a shift between this alpine taxa to this alpine subalpine taxa. So you can see how different groups of altitudinal vegetation appear along time. And you can also see a rise in richness as long as those species appear in our studies in our lake. When we get to the start of the Holocene, you can see a predominance of this subalpine taxa that has occupied the habitat for the former communities, but also you can see this notable rise of temperature, and also this decrease of meadows that prompted a decrease of richness. Then, as we approach to the Holocene climatic optimum, you can see that more species, more communities or species groups appear to our lake. We have subalpine, montan, and montan. We have this kind of communities there. And also some influence of choline taxa. You can see also that you have the establishment of the woody taxa in our place. So our lake landscape completely changed. This prompts a decrease of richness, but also an increase of more organic matter gets to the lake. So you have a higher amount of biomass surrounding this lake. As we get to the early Holocene, you can see that you have some climatic fluctuations that makes your montan taxa disappear. And then only this subalpine and subalpine montan taxa continue there. And you can also see an increase of the forest that makes a decrease of the total richness. If we get to the last 3,000 years, you can see that forests continue, like you can see kind of an expansion of the mixed forest, and richness continues more or less stable. But you can also find another influence of taxa coming from choline and montan taxa. So it would be more lowland taxa. And in the very last part, which covers the last 1,000 years, you can see that two different communities appear, and they have two different, very different altitudinal zones. In one side, you have this alpine subalpine taxa, that would be this one. And then you have this influence of montan choline and even choline, that would be this one. Also, you can find a huge increase of the total richness and very marked deforestation. So at this point, you only have meadow, graminoids that are a very small amount with this marker, and shrubs. So now I want to go to humans. And this is just a transition slide, but what I want you to see here is how we use different proxies or indicators and how they match between them. In this part, you can see the coprophilous fungi, which come from the feces of animals, and the pastoral indicators that we found with the autocology of species from our sequences. And the trends are quite similar. And then, if you look at the arrayables, this is the ones that we got from our DNA sequences. And these are the pollen frequencies from the very same lake. And they are also telling you the same story of presence. So now let's plot this in a different way So we can see the erosion indicators, arable indicators from DNA, the charcoal, meaning fires, and the pastoral indicators from DNA and coprophilous fungi. We also have the temperature, the total richness, and the Forbes richness, and the functional groups, all according to age and depth. Also, I find that here it's very visual to see that in the moments where you have a decrease of richness, even the total or Forbes richness, you have an increase of woody taxa, even if they are shrubs or trees. And in the opposite way, when you have a higher amount of richness, which is also very correlated with the Forbes richness, you have a dominance of the meadows in your place. Let's look at the human influence and grazing influence. The first grazing influence that we can infer from the coprophilous fungi are around 8,000 years ago. And you can see that this coincides with a rise of richness. The second most marked event comes with the Neolithic, with a high erosion in our catchment, and also the introduction of the pastoral indicators that we find with DNA, a part of the coprophilous fungi. And you also have an increase of richness, and it co-occurs with a higher meadows presence. However, if you have a look to the Forbes, you can see that you have a decrease of Forbes, which means that you have a higher amount of meadows, but they are less diverse. If we continue, we have to go to the late Bronze Age, when we have a higher erosion, but also we have the introduction of arable indicators, which means that close to our catchment, there were some crops or some agricultural practices, which doesn't mean that it was directly in the lake, but yes, on the surveillance of that lake. We also have fires that talk about the landscape management that humans did in our lake. It's different from the past. And of course, we have pastoral and coprophilous fungi. This is also related with an increase of richness and an expansion of meadows. And with the Middle Ages, we can see a high amount of fires with the same agropastoral activities. But if you look at the total richness, you can see a really high and short increase in richness in a very short time. This is also related with the deforestation of Sulsoli, where trees almost disappear. And finally, with the Industrial Revolution, we can see the higher grazing effect in our lake. And another rise or slight rise in richness. With that, I will go to some conclusions where we can say that temperature has been the main driver of vegetation during the first half of the Holocene. But as long as we get to the Neolithic, humans and climate has shaped the landscape of Sulsoli. And humans has influenced it through fires and land use management. And this has also changed a long time, and also the pressure of their impact. What we can also see is that the agropastoral intensification that occurred during the medieval age is the one that prompted this huge rise in richness that nowadays is characterizing the diversity of this site in the Alps. And what we have also seen is that the high taxonomic resolution of cDNA that we could get using a regional database for the Alps, and also the availability of the autoecological data has allowed us to define reliable indicators that have allowed us to perform robust ecological reconstructions from the cedar DNA of this lake. Finally, I would like to have a very short reflection. And it is I don't know if you're familiar with that. This is a human castle. It's very typical from Catalonia, the region where I am, but it matches a bit the idea that I have. And it's that the interaction of this, so you need several steps to get to these reliable cedar indicators. We need the interaction of several disciplines that has allowed us to achieve this. And they have been very important for that. First, this DNA metabarcoding knowledge and proper methods of cedar DNA in the lab, together with robust bioinformatic pipelines, has allowed us to get to this level. But also this complete regional reference database that allows us a high taxonomic resolution. To get the taxa that we could compare with the topological data, all these disciplines together allow us to get to these reliable cedar indicators to do proper and more robust paleoecological inferences. With all this, I would like to thank you a lot for your attention, and also thank to all the people that has made this study possible.
Thank you, Sandra, for the very interesting talk and for bringing us so far back in time and letting us know what our human impact has done on the landscape. Very fitting for today's theme of day. So I would like to ask if anyone here has any questions. You can either type it in the chat or raise your hands using the reaction button. Okay, since nobody has a question, I have one. So with all the data that you have collected, I'm just curious to know if it is possible whether you can correlate these changes in plant diversity due to direct competition of these species with each other rather than the effect of temperature or human, for example.
Yeah, this actually there are some parts when you have less human impact, when you can find some biotic interaction. So as I said, these altitudinal stages are mainly driven or shaped by temperature, but this is for the higher part of this zone. The lower part of the zone is mainly shaped by competition between both zones, the communities of both zones. So what you can see is that, for example, you have an increase of temperature, then your montane taxa, the lowland taxa, is going higher. And this one, it's competing with the taxa that is present in that lake. So there is a competition effect that you can see there. Also, in that zone where you have an increase of shrubs, you can see how shrubs compete with forbs. And that creates an homogenization of the landscape that reduce the meadows and the richness.
Okay, thank you for the answer. So if anyone has any more questions, you can just type it in the chat and direct it to Sandra. And now we'll move to our last speaker of the day. I introduce to you our keynote speaker, Dr. Tyler Kartzinel. Tyler holds a PhD in ecology from the University of Georgia and completed his postdoc at Princeton University and the Nature Conservancy. He is currently a Peggy and Henry D. Sharp Junior Assistant Professor of Environmental Studies at Brown University. Research in his lab focuses on conservation biology, and much of his group's research uses molecular tools and field experiments to illuminate how species interact with each other. Included here is a link to his website if you are keen to find out more about his research. So now I'll hand over the time to him.
Thanks so much, Physilia, and thanks everyone for coming out and organizing such an exciting seminar series. It's really fun to be able to participate remotely. So I'm going to talk about the trophic ecology of large herbivores today. And the focus of my talk is going to be on understanding animal diets and strategies that we have to do this. And I'm going to emphasize dietary DNA metabarcoding, especially in African large herbivores for this talk. Dietary DNA metabarcoding is a molecular strategy to identify what animals eat. And so our protocol begins with animal dung collected fresh from the field. That dung contains undigested plant cells and maybe some digested plant cells from which we can extract plant DNA, sequence it, and match it with a reference library of known plant species. And when we see an exact match like this, we know exactly what an animal has been eating over the course of the previous day. And practically on the computer, what we get at the end of this is a really large data set that includes a list of Latin binomials, plant species that animals have been eating, together with information on the frequency and the relative abundance of those plant species and animal diets. Most of the data I'm going to present today are based on one marker, TRNL-P6, which I've heard other people talk about today. But that's not the exclusive type of data that we have available. I sort of took some inspiration from the name of this particular symposium, Proxy Wars. I thought that was a really clever way of framing a series of talks. And so I wanted to offer a sort of provocative battle-hardy statement that I've been thinking about a lot and reflecting on, which is animals do not eat Latin binomials. And this is not a new idea. Dan Jensen penned this in 1979, but it's one that I think people don't expect me to offer to share, especially in light of the method that I just described, which is known for generating lots of information on Latin binomials. And so I want to sort of guide you through this talk with this thought in mind, and I'm sort of going to explain to you what I mean by this, the tension that exists between our ability to generate information on Latin binomials and our desire to understand the ecology of herbivores and what they eat. In ecology, there are really two types of comparative dietary data in wide use today. One on the left, I sort of call a static network. It's sort of the classic traditional way of drawing a food web. There's consumer species and resources, and they're connected through an interaction network. Now, this is the sort of framework that most dietary DNA metabarcoding is cast in. It's consumer species connected to resource species. On the right, there's another type of data that's in wide use. And I sort of call this a trophic agglomeration, where we often know what the consumer species are, but we don't really know what the resource species are. We think of them as sort of a gradient, of different resource types. In herbivory, in herbivores, we often can think of this as information we gain from stable isotopes, which won't tell us, what species an animal is eating, but might tell us the type of plant it's eating from. In predator-prey systems, it might be a size distribution of prey items, big versus small. And these two perspectives together let us make important, really generalized statements about food webs, but I think that they sort of limit our ability to understand the fast pace of foraging decisions that animals are making. And these limits, I think, can be overcome using dietary metabarcoding. Neither is inherently better or worse than the other. They're sort of complementary, but each has limits that we want to address here. So we're going to do this by taking a close look at how these perspectives play out in a system of herbivores, large herbivores, at Impala Research Center in Kenya. This is a 200 square kilometer unfenced wilderness with a nearly full complement of large herbivores and predators. It's a semi-arid savanna that looks like this. There's a sort of broken canopy dominated by acacia trees, and an understory that's more or less continuous grasses and forbs. Together with all of the wildlife, there is an active ranching operation here. So there's herds of cattle that are tended and maintained in ways that sort of mirror what the traditional pastoralist practices in the area look like. Now, in these types of savanna ecosystems, ecologists have long sort of imagined really two different types of plant species and thought that herbivores would partition their use of these plant species in ways that help them coexist. We call this a grazer-browser spectrum, where on the left here, there might be some species of herbivores like zebras that eat mainly grasses. And on the right, there might be antelope that eat mainly trees and shrubs, other species. And in the middle, there's a group of animals that we call mixed feeders. Their diet comprises a mix of grass and browse. And by partitioning their use of these two different resource types, they sort of mitigate competition and are able to coexist. Within these two broad guilds of browsers and grazers, we also know that animals partition their use of plant parts, and this can help enable coexistence. And so, classically, giraffe have really long necks. They're able to access forage that smaller herbivores can't access. They don't have to compete, and that contributes to their ability to coexist. Same thing happens in grazers. Large herbivores tend to eat from taller, less productive grass swards, whereas smaller grazers tend to eat short, very productive grassly lawns. And the idea is the same. By partitioning their use of these different types of plants or plant parts, these herbivores reduce competition and are able to coexist. It helps explain the great diversity of wildlife that we see in ecosystems like this. Now, about 10 years ago, we began to realize that these sorts of simplistic approximations may not actually practically help us explain coexistence. And that's for a couple of reasons. One is that many similar species of grazers and browsers often coexist. We observe them together in nature. And so this is a picture of zebra grazing on a lawn. Actually, there are two species of zebra here grazing together in the same place and at the same time. The zebra on the right of the screen with the stripes that go all the way around the belly, those are common plain zebra. They're distributed all across African savannas. On the left, the zebra with the bare belly, that's a Grevy's zebra. They're an endangered species. There's only about 2,000, maybe 3,000 of them left in the world. So we see things like this all the time in nature. The other problem is that grass and browse comprise enormous taxonomic, functional, and phylogenetic diversity. These are hugely diverse groups of plants, and there's diversity contained within them. So we can reason that herbivores might partition their use of plant species, and that could contribute in addition to strategies that help them coexist. We could hypothesize that herbivores partition dietary plant species according to body size. So they're not just accessing different parts of the same plant species. Large and small herbivores might be accessing different suites of plant species altogether. We could hypothesize that herbivores of similar size eat different suites of plant species, even when they're in the same parts of the spectrum. So two zebras that eat similar proportions of grass may be focusing on eating different grass species. To facilitate this work, we've built a really extensive plant reference library for the ecosystem. The protocol here, we begin with sampling live plants out in the field. We voucher them and archive them in relevant major herbaria. We sequence reference plant DNA and contribute these DNA sequences to online public reference libraries. To date, we've sequenced, we've generated about 6,000 barcode sequences, and those are from nearly 2,000 specimens. And they represent about 500 plant species, which is close to what we expect for all of the plant species in the region. So we can do a really good job identifying plant species and animal diets in this way. And this is a quick talk, so this is going to be my one slide dedicated to the plant reference library, but I just want to add on a personal note. This was a lot of fun and a lot of effort to build. It's been an effort going on for more than 10 years. Every time we do a new bout of sequencing and analysis, we learn something new, not just about the plant diversity in the region, but what the animals are doing together with those plants. When we released the most recent version of this database, it increased the size of public barcode data for plants from the entire continent of Africa by 10%. It's an enormous contribution, but it's also, honestly, a really modest contribution. This is 6,000 sequences, a couple thousand specimens compared to what the major world-leading institutions are able to do, this is a few days of work, right? You know, for me as an ecologist, it was a lot. So, you know, I just want to sort of offer, you know, to those people who are maybe in charge of such institutions or influential in these spheres, partnering with African institutions, African scientists, and contributing to this type of work is one of the most meaningful and impactful things that we can be doing. It doesn't take a lot to have a really big, important, meaningful contribution. So getting back to the dietary analyses, you know, one of the first things we wanted to know is, can this DNA metabarking approach be both reliable and quantitative? So, you know, at the very beginning, we focused our analysis on 7 species, the 7 shown here. These species represent, you know, the entire range of the grazer browser spectrum. On the left, we see the 2 zebras that I've already introduced. We know that they eat mostly grass. Grazing cattle and their wild analog, Cape buffalo. Then in the middle, we sort of have mixed feeding impala, common antelope, and the world famous elephant. And all the way on the right of the screen, we have a pure browser, an animal that we know eats almost no grass. It's a 5 kilogram antelope called a dik-dik. It's about the size of a house cat. So compared to, you know, a 5,000 kilogram elephant, we have, you know, the biggest and smallest herbivores in the system as well. What we found by comparing the relative abundance of grass DNA in samples from these animals with corresponding measures of grass and diets from carbon stable isotopes, sort of the gold standard reference point that we had, we found that DNA metabarcoding can be both reliable and quantitative. We have a near perfect one-to-one correlation. There's some variation around that one-to-one line, but it's really good benchmark. And what we also see is reflected in this ecological reality. We see the grazer browser spectrum. Yeah, they're the dik-dik on the bottom left. They eat almost no grass by either measure. Zebras, top right, and an elephant and impala, the mixed feeders, they fall somewhere in between. We found that dietary differences increase with body size disparity. And so what this shows is, you know, a difference in body size from all of the herbivores in the study, together with information on dietary overlap or dissimilarity. Close to the sort of vertical axis, we see 2 similar sized animals, the Groby zebra and cattle. They have relatively similar diet composition compared to the 2 most dissimilar in body size animals, the dik-dik and the elephant. So this suggests that animals of different size are not just eating different parts of the same plant species, but are accessing different suites of plant species altogether. And here we have an ordination showing the dietary composition of nearly 300 samples from each of these species. You know, it's a cartoon that simplifies the complexity of the system. Points that are close together represent similar diets, and those that are far apart represent very different diets. And they're color coded according to herbivore species. What's amazing about this to me is that along the horizontal axis, we see the grazer browser spectrum, the one that we always expected. We see zebras, specialized grazers, cattle and buffalo to the right of them, mixed feeding elephant and impala to the right, and dik-dik, the pure browser all the way on the right. But what separates this from what we've been able to see before is the vertical dimension, where we can see animals that eat identical proportions of grass. We'll have some separation. There's some dissimilarity in their diets that we wouldn't have been able to appreciate previously. So to sort of zoom in on this, we, you know, in a comparison of the 2 most similar species, the 2 different species of zebras, we can compare foods that differ in frequency in their diets. 45 plant species were commonly detected in these animals' diets. 30% of them were shared in roughly equal proportion. 5 of them were more often eaten by the plain zebra and 10 more often eaten by the Groby zebra. So, you know, together, fully a third of the plant species present in these animals' diets distinguish their diets, right? So ecologists haven't been wrong all this time. You know, it's been correct to say that zebras eat grass and mostly grass, and even many of the same species of grass. But what we're beginning to appreciate now is the magnitude of the difference. One of my favorite stories from this work has to do with an instance of molecular science sort of rediscovering local knowledge. So here I'm showing, a cattle grazing on a grassy sward and a legume, a small fabaceous plant called Indigofera stederi. And when I first got a version of these data, I found DNA from this plant in basically every cattle dung sample that I analyzed. And in the literature, we think that cattle are mainly grazers. They eat grass. Right? And that's what agriculturalists think anyway. And I was in a panic. I was a postdoc at the time. I was thinking all of my samples were contaminated. This must be an error. But when I did a little bit more research, I learned that pastoralists, the traditional herders in the region, have actually long understood that this plant is really important. They attribute it to the high quality of cattle growth, high quality of cattle milk, and they actually time their movements with their animals across the landscape to match the peak productivity of this particular plant species. So this was, a really sort of fun insight to gain and an opportunity to connect, our sort of modern molecular approach to science with, more traditional ways of knowing. One of the advantages that we have from using molecular data to understand animal diets is that we get to begin exploring relatively unexplored aspects of plant species, clades, and traits in animal diets. And so here I'm showing a kind of complicated, colorful figure, and I'm not going to ask you to remember everything about it. But to orient you, what I'm showing is the diet profiles of all 7 of these herbivores vertically. And I'm matching it with a plant phylogeny to the left. What we see vertically then is the diversity of species that these animals are eating, where each of these colorful bars represents the presence and frequency of each of these plant species in an animal's diet. But what we see in this dimension from left to right is a representation of phylogenetic and trait diversity that we haven't really been able to access and appreciate before. Down here in purple, we have Poaceae, that's the grass family. And what we see is that the 2 specialized grazers eat a lot of grass, right? Just as we've always known. Here we have Fabaceae, that's the legume family that includes acacias, the dominant tree in the region, but also those indigoferas that I just described, as well as some other common plant families from these different lineages. We see that grazers specialize on one plant lineage. Browsers eat from many different lineages, but really don't eat much grass, and mixed feeders fall somewhere in between. Now, as our data set has grown, we've been able to sort of compare the taxonomic and phylogenetic dietary diversity for 25 herbivore species from this area. And this graph shows, these 2 graphs show on the x-axis for both a broad diversity of ruminants and non-ruminant animals, the relative abundance of grass in their diets. And on the left, we're showing the taxonomic diversity. So specialized browsers and specialized grazers have relatively low dietary diversity compared to mixed feeders that are able to get food from both of these different plant groups. But over here on the right, we're showing phylogenetic diversity. And what we see is that an animal that eats more than about 20% grass quickly has the phylogenetic diversity of foods in its diet plummet. So there's a decoupling of taxonomic and phylogenetic diversity. This is a little cartoon that shows how we're thinking about this. And the upshot is that taxonomic specialists can be phylogenetic generalists and vice versa. So here we're sort of plotting taxonomic rank, the taxonomic specialists and the taxonomic generalists, together with the phylogenetic rank, the specialists in terms of the phylogenetic diversity in their diets and the generalists. We see that there are some species that specialize in both ways. Zebras, for example, don't eat too many plant species compared to the others in their ecosystem. And they're drawing their diet from a very narrow portion of the phylogeny. But there are some grazers eating similar amounts of grass that actually eat a whole lot of different species of grass. And the same can be said about the browsers. Browsers eat, a broad swath of the plant phylogeny that's available. But there are some species like camel and kudu that concentrate on a relatively small number of plant species. They're just drawing those plant species from different lineages. And you can compare that to impala and eland, and you see that there are generalists in both dimensions as well. So a crucial take-home message here is that precision alters perception. Recognizing this sort of compels us to challenge assumptions. Data show that we could conclude diets are mostly redundant based on proportions of grass that animals eat. This is the grazer-browser spectrum. But we can also conclude that diets are largely unique based on differences in the food plant species that animals are eating. I think both of these perspectives are true. We've just historically focused much more on the first idea. So I want to pause here to ask ask for help avoiding common misconceptions about the ways we should interpret these data. We can't assume that any of these animals we're sampling are eating the preferred food species. They're making do with what's available, right? And we can't assume that diet partitioning alone is evidence for or against competition. Right? It's tempting to conclude that animals that don't eat too much of the same types of foods aren't competing. You know, they perhaps evolved. You know, there's been character displacement that facilitates a minimization of competition in this way. But competitive displacement is also a thing. Right? So animals could be competing fiercely and excluding one another from resources that they prefer, and that could be reflected in these data too. This leads to new research priorities. We want to know how variable are individual diets, and when do these differences in food identity represent differences in nutrition behavior or competition, things that matter for our understanding of biological diversity. We need detailed field studies and experiments to do this. And so in the couple minutes that remain, I sort of want to just share a few unpublished results that sort of hint at the ways that we're trying to accomplish this in my research group. How do the diets of individual animals vary through time? You know, they live in variable environments. To get at this, we're sort of looking at the foraging history of individual elephants in one of the best studied elephant families in the world, the elephants of Samburu in Kenya. And this is a very seasonal environment. So you can see up here, you know, at the top from a period of 2000 to 2005, rain fell in bouts, and each one of these bouts caused a green up, a big flush of plant productivity, primarily grasses. And predictably, the elephants began to eat grass whenever this would happen. So you can see, based on carbon stable isotopes obtained from the hairs on their tails, which are incorporating molecules from the foods that they eat, that there are these corresponding peaks. Every time there's a green up, they shift their diet to grass, and you can see that in the isotopes in their tail hair. This is something that's been known for a couple of decades. We thought we'd try to see if we could do it using metabarcoding as well. And so now we have fecal samples from 2 individual elephants in the same family group through time, through some of these seasonal pulses and productivity. And we expect maybe 30 or 50% grass in their diets at these peak moments based on isotopes. These are data representing the plant families that we sequenced from these animals' diets through time. And you see a really complex picture. The colors represent different plant families. We see grasses here in purple peak periodically, but they're never more than 30 or 50%. The acacia trees and other legumes, that's a big swath of it. But you can also see that there's other plant families in here playing interesting and dynamic roles. And you can see that these 2 elephants from the same family, living together, eating from the same swards, eating from the same forests. There's not much correspondence between what they do, right? So we can see that members of a family group select different plants at the same time and in the same place, and that's really interesting. How do elephants compete with other tall species, like giraffe? Elephants are struggling in many parts of their range, but in Kenya, they've been protected, and so the elephant population has actually grown a lot in recent decades, and conservationists are concerned that they're competing with other endangered species, and so they're building these big experimental fences that keep elephants away from other herbivores to try to provision the herbivores with good food. Expectedly, when you go inside some of these exclusions in the absence of elephants, trees become more abundant, taller, and more accessible. This is the provisioning of giraffe food for giraffes, and this is how it works. Excluding elephants, we can see alters giraffe diets. So here in the background, I'm showing individual giraffe diet profiles in the presence of elephants and in the absence of elephants, and you can see how strikingly different they are. In the presence of elephants, they have about 40% acacia food in their diet, nearly doubles in the absence of elephants. That's what, you know, these big orange blobs are, you know, closely related acacia trees. If we think about this in terms of what foods are available and compare, you know, diet, what they're actually eating with what's available, we can see that in the presence of elephants, these giraffes are working really hard to get that 42% of acacia. They're positively selecting it. But in the absence of elephants, they're getting nearly double the amount of acacia in their diet, their preferred food, but they aren't working as hard for it. They're sort of like looking for other things to eat now as well. Evergreen shrubs, less palatable food plants. Giraffe are pretty ambivalent about it. Their selectivity is just about zero in either environment. Right? So now we're seeing differences in what animals are eating through time and through space, and we're able to sort of experimentally try to understand why. You know, how much of this variation might we attribute to competition? So the main results here, to sum up, are that diets are not immutable properties. Adding and subtracting species from ecosystems changes networks. And these changes reflect variation in both the food availability and individual foraging behaviors, what the animals are actually looking for. So we learned that we miss something if we assume the uniformity of species, like we would in a classical food web study. And we also miss something if we accept the anonymity of species as we would in an isotope study, for example. Neither of these approaches is telling us something wrong. They're just complementary. They're telling us different parts of the story. Going forward, I think it's going to be really fun to think about how different parts of the same plant species differ in quality and quantity. So here's an imaging spectroscopy view of an acacia tree. The bright red coloring here represents nitrogen, chlorophyll, high quality foods. And you can see even on the same plant, you know, and it distributed across the environment, the quality of food that animals have available will differ. And we want to understand how this influences what they eat. We've barely begun to incorporate predators into these types of analyses, and these animals could have even stronger influences on trophic ecology than competition. So that's another really exciting future direction to take this work. To sort of sum up, our field is really on the verge of some breakthroughs. We can now use this technology to split and agglomerate food taxa according to phylogeny. We're not constrained by which perspective we take. We can characterize dietary variation within and between species, and we can repeat this through time. And that opens so many doors for us. And this matters because these are dynamic systems operating against the backdrop of change. Here we are on Earth Day. This is a simulation of what vegetation is going to do in Kenya over the next 30 or so years. We're expecting a major drought, major dry down, and lack of productivity. We know that these animals are going to have to change their foraging ecology. So Latin binomials are useful proxies for multiple dimensions of diet. And over the past decade, we've become really good at using this type of molecular approach to infer Latin binomials. But in the decade ahead, I'm really excited for the ways that we're going to use these tools to understand ecology more broadly. So thanks. Yeah, thanks. And looking at my clock, I ran a couple of minutes over, but if there's time for questions, if folks want to stick around, I'm happy to chat.
Yeah, thank you, Tyler, for a very interesting talk. I learned so much more about animal competition, and because I'm also studying feces, diet from feces, so it's quite a good research. Do anybody here have any questions for Tyler? Okay, Barbara.
Thank you very much for a very interesting talk, very impressive results. I was wondering, do you also study invasive plants? Because now you mainly talk about a native plant species, but as you know, that the world is increasingly distributing plant species intentionally or non-intentionally. And in my part of the world, herbivores also either eat or not eat these species. So could you say something about that? How static are these diets? That's what I'm meant to ask.
Yeah, absolutely. It's a great question. And it's one that, you know, I'm really turning my attention to now. So in many of the temperate grasslands, for example, it's the non-native species that are sort of invading and bringing in photosynthetic diversity that wouldn't naturally exist in the grasslands of North America and the steppe, for example. That are the most successful invaders, and they're changing phenology. They green up earlier and perhaps brown down at different times, and that influences probably where animals are going. We know that green up and green down timing has historically influenced where animals are going, and there's no reason to believe that a change in that phenology wouldn't have an impact. So this is, especially with climate change accelerating and the redistribution of species around the world, a huge important example. There's also, we recently with collaborators published a really cool study of one of the 100 worst invaders, plant invaders in the world, in a study from Gorongosa National Park in Mozambique. And so there's this shrub species called Mimosa pigra that invades wetlands around the world, and there was a sort of recent defaunation event in this national park, and the plant invaded, established, and really took over. With restoration, there was a question about how do we restore this ecosystem? Do we leave this plant here, or do we at great expense beat it back with burning, cutting, pesticides, things like that. And what we found, with a little bit of study using metabar cutting, is that actually the native herbivores were eating a lot of it. And so as those populations were recovering, they naturally reestablished some control over it. They suppressed it. And so it wasn't like this non-native invasive plant species was choking out the native plant species that the herbivores would have preferred to be eating. It was actually fueling the regrowth of the native herbivore populations that were naturally controlling it. And so, there's great opportunity to test cool ideas in basic biology using invasive species, but there's also, opportunities to really engage with conservationists and restoration biologists to think about how we can use these tools to guide decisions that they're actually making on the ground. I really appreciate that question.
Thank you very much. Very interesting.
Do we have any other questions?
Tyler, yeah, I had the same question as is in the chat from Naziz. Which marker did you use for your studies?
Yeah, most of these data that I presented today were TRNL P6. In the system where I was presenting, we can distinguish about 80% of the plant species in the local flora using that marker. But of course, there's some grasses and acacia trees that it's just a whole bunch of species that we can't distinguish. So
80%. I wouldn't expect that, but that's.
Yeah, no, it's high, and I think it's high in part because of the type of ecosystem which has representation of plants from many different lineages. It's those more recent sort of diversifications, the grasses in particular, but also the acacias that are abundant and dominant in the ecosystem that we still struggle to differentiate. We've played with ITS and more taxon specific markers, and those data are out there and published if you find them interesting.
Common for everyone, I guess.
Yeah, I would really love to and have plans to experiment with longer markers, to try to do a better job. But there's so much work happening there now that I'm, as an ecologist, sort of watching the trends and trying to figure out what smart and engaged people come up with and learn from them.
I think we have time for just one last question, if anybody has one. If not, I have one. So you spoke about building reference library, and I was just wondering if there are any plans in the future to sequence whole chloroplast genomes or ribosomal nuclear genomes instead of just sequencing barcode regions.
Yeah, Active plans, no, unfortunately, but an interest and desire, yeah, absolutely. We have the DNA from this barcode library. We're building barcode libraries in other systems in North America and other parts of Africa right now, very actively, and starting in South America too. So yeah, for all of these projects, for boring practical reasons of time and expense, we're going to keep focusing on barcode markers. But you know, if there are people who are interested in getting more information, who maybe have the wherewithal to know how to use it, we'd be very keen to collaborate. It's the more information, the better. You know, even with like a full battery of typical, you know, quote unquote standard barcode markers, we still lack a barcode gap in some important plant species that would like to be able to distinguish, and having a full chloroplast genome would give us more options.
Okay, thank you, Tyler, for your talk. So before we go, I have just a small reminder that the continuation of our seminar series will take place again on the 27th of May, which is on a Thursday, where the theme would be on illegal plant trade, and we hope to see many of you again. So thank you for joining us today. But before you go, we would now like to take a group picture to commemorate this seminar series. So please leave your video on if you're comfortable. If not, you can leave it switched off. And Marcella will be the one taking the picture.
Just seeing if a few more people are turning on their cameras or not. Looks like it. So, yes, indeed. All right, then. All right, everyone's looking. All right, 1, 2, 3. All right, thank you so much, everybody.
Thank you for your time.